I don't have anyone named iPhone 57 on my class roster.
It's Cofield, Acacia.
Acacia, I was just kidding.
Oh.
iPhone 57.
All right, we have one chapter worth of material left to go today. It's a small one. We're talking about lipids, and lipids are sort of a catch-all category of molecules. All right. They're not polymeric, which means that we don't have like single amino acids coming together to make a long chain called a protein. We don't have nucleic acids forming, or sorry, nucleotides forming a long chain that we call nucleic acids. We don't have a bunch of monosaccharides linked together through glycosidic linkages to make a polysaccharide. Lipids are small. All right. So the key with lipids is they are a type of biological molecules that don't dissolve well in water. All right. So they are non-polar compared to all of the other types of biological molecules, which they tend to be pretty polar. All right. They don't have a particular functional group. Depending on the structure of the lipid, they could have any one of a number of different functional groups in them. We'll talk about the functional groups that they have in them today. And they have lots and lots of hydrocarbon in them. All right. And we know that that means since they're not very polar, they don't dissolve well in water because we know that like dissolves like. All right. So we can talk about lipids as a type that are called hydrolyzable. So hydrolysis is a kind of reaction that we've talked about a bunch of times. Like, for example, we have learned that amides and esters as functional groups can be hydrolyzed. They can be split back apart. So an ester can turn back into its carboxylic acid and its alcohol part. An amide can turn back into its carboxylic acid and its amine part. All right. So lipids that are hydrolyzable contain some functional group that can be hydrolyzed. All right. Generally speaking, they're ester functional groups in the lipids that can allow them to be hydrolyzed. So we'll look at some examples of those. There are also some non-hydrolyzable lipids, all right, ones that even if we were to treat them with a pretty strongly acidic solution, there is no functional group in them that can be split apart in those conditions. So the big one that we're going to talk about are the steroids and, like, more specifically cholesterol as an example of a non-hydrolyzable lipid. So the hydrolyzable lipids all have one thing in common, and that is that they contain these things called fatty acids. All right, fatty acid. It's a really easy term if you know organic chemistry to the extent that we do now. Fatty means that it has a really big nonpolar part, and acid means that it contains a carboxylic acid. All right, when we look at the structure of one such fatty acid, you can see why it gets its name. All right, carboxylic acid functional group. That's where our fatty acid comes from. And then the fatty part. So fatty is like another word for nonpolar in this context. So fatty acids contain a polar functional group at one end and then a really big nonpolar tail. Note that these fatty acids formed the basis of what we made into soaps early in the quarter. All right. The only difference is we had them in the form where the carboxylic acid was deprotonated instead, making the head group even more polar than it would have been before. This is not something I'm going to test you over, but it is interesting. So you know how we just see these things that naturally happen in the world of biology, even though there's There actually is a great explanation for the even number of carbons in the fatty acids. I don't think there is a great explanation for why we chose to use the L versions of the amino acids or why we use the D versions of our monosaccharides and not the other version, but we do. It's just an observation, all right? There is actually a metabolic reason behind this. So fatty acids tend to be metabolized 2 carbons at a time. So it makes it easier to metabolize them if we have an even number of carbons. All right. Just a little bit of language. You guys have heard of the term saturated fats before. All right. We are going to make sure you understand what that means now. And that is that saturated fats have fatty acids that don't have double bonds anywhere in that long hydrocarbon chain. All right. So unsaturated fats are fats where the fatty acids have at least one double bond in that long chain someplace. Now, another really interesting thing is to do with the stereochemistry of any double bonds that are in those fatty acids. It turns out that when we have double bonds in our fatty acids, they're always the cis version, not the trans version. All right. It turns out that our enzymes are really good at metabolizing cis double bonds, at least in fats. And either they metabolize trans fats really slowly or not at all. So when you read about the potential harms of trans fats generally are not natural. They're things that humans have made, all right? The same way that we can modify or create an artificial version of sucrose called Splenda, where some of the hydroxyl groups are replaced with chlorine atoms, all right? We can create artificial versions of fats called trans fats, where the double bonds are trans double bonds instead of cis ones.
Is that the internship which gives many people gastrointestinal distress, trans fats?
Trans fats do, yeah. So the classic example of this is a commercial product called Olestra. I don't know if you guys have heard of that before. You've heard of it before, Nicole? Mm-hmm. Yeah. So Olestra is a type of artificial fat that was essentially non-caloric because the body can't process it. For example, they used to sell non-fat Lay's potato chips that had Olestra instead of natural fat as the thing that made the Lay's potato chips taste particularly greasy and delicious. All right. Or non-fat Pringles, I think was an even more popular one. So it turns out that when you market something as non-fat, consumers tend to eat more of that thing in a sitting than they would of the stuff that contains the fat. So a lot of people ate, like, for example, an entire can of Pringles at the same time, which is a huge ingestion of this unnatural fat that your body cannot process. So if your body can't process that fat, it just goes right through your GI tract and comes out the other end. If you have a lot of fat coming out the other end unchanged, you get symptoms like oily diarrhea. Yeah. Yeah. Severe symptoms. So it turns out that the more double bonds we have in our fatty acids in these lipids, the lower their melting points are. Now, what that means is that it's not a hard and fast rule, but we know that unsaturated fats tend to be healthier for us than saturated ones. So think about a way that you could sort of very quickly tell the difference between a healthier and a not as healthy fat. One of the easiest ways is if that fat is a solid at room temperature, it's probably not as healthy as one that's a liquid at room temperature. All right. Which is healthier for us, olive oil or Crisco? Olive oil. Which one is a solid at room temperature? Crisco. All right. So it's not a perfect way of telling, but generally speaking, fats that are liquids at room temperature, so we call those oils instead of fats, those oils tend to be healthier for us than the more saturated things that tend to be solids at room temperature.
So then why is there so much hype on the coconut oil?
Coconut oil isn't actually particularly healthy.
That's what.
It has
but-
lots and lots of saturated fats. So we'll look at the distribution of the different kinds of fats. Coconut oil does happen to have some things about the fatty acids in it that are particularly good for certain kinds of diets. So there's a lot of hype around something called medium chain triglycerides. So that is a triglyceride is a kind of lipid with 3 fatty acids attached to it. Medium chain means that the hydrocarbon parts aren't really long or really short. So those happen to help induce or maintain ketosis. So people on the keto diet really like having lots of medium chain triglycerides in their diet. And coconut oil has lots of those. But coconut oil is mostly saturated fat. And general dietary recommendations say we should limit the amount of saturated fats in our diet. Yeah,
Chemistry perspective.
right.
because
So
of
people think
nutrition,
that,
She said the same thing, it's not all that healthy.
yeah, it's
because
not.
It's a solid.
It's not. It is really good for your hair though.
Yeah, we've been using it in our skin care for.
On your skin? Oh my God, I would break out so hard. Like any oil on my skin. No.
I keep it.
I have a friend who's so sensitive that the only thing that he can do to wash his face is like just hot water in the shower. Can't even, no soap, like no product, nothing. Like rinse the extra oil off with hot water and then hope it takes care of itself. Other than that, and he has good skin now, but never did before he started just the hot water wash and nothing else. All right. So let's look at some examples here. Stearic acid. You don't have to know how to name these fatty acids, at least not the common names of them. All right. So stearic acid, we could count the number of carbons. I actually don't remember how many carbons stearic acid is supposed to have. That should tell you something. If I don't remember those names, then you don't need to either. All right. You can see oleic acid is different because it contains a double bond in that hydrocarbon chain. Now, cis double bonds, one of the things that we know about them is that a cis double bond changes the direction of the hydrocarbon chain. You can see that, right? That actually is really important in the world of lipids. So the amount of unsaturated versus saturated kinds of fatty acids that you have in your lipids actually regulates some really important things. Like when we talk about the phospholipid bilayer, the amount of unsaturated fatty acids in that bilayer actually changes how much the components of the phospholipid bilayer can move around based on how tightly they pack. Super interesting. So here are some common fatty acids. Notice that over here in this column, the number of carbons, all of the natural fatty acids have an even number of carbons in them. All right. So the easiest way to explain that is based on the fact that we metabolize fatty acids with even numbers of carbon much, much more easily. But it's true. You will not be responsible for knowing any of these common names of the different lengths of fatty acids. It's too much. Now, what you will be responsible for is understanding the convention that we use for naming fatty acids that have double bonds in them. All right. So in a fatty acid with a double bond, because these chains of carbon can be really long, like up to 20 or more carbons, and because the carbon-carbon double bonds tend to come toward the end of the chain instead of the beginning where the carboxylic acid is, we've developed a naming convention for talking about where the double bonds are that starts the numbering from the last carbon in the chain instead of the first carbon. All right. So if you have ever studied the Greek alphabet, the very first letter in the alphabet is in the Greek alphabet alpha, last letter in the Greek alphabet, omega. All right. So when we talk about the alpha carbon, we're talking about like the first one next to whatever functional group we're talking about. We're talking about the omega carbon. We're talking about the one furthest away from the functional group. So if the carboxylic acid in fatty acids is the important functional group, if we talk about numbering something based on the omega position, we're talking about numbering starting from the furthest carbon away from the carboxylic acid. So let's look at an example of what that looks like. Here's our carboxylic acid in linoleic acid. Instead of numbering all the way from carbon 1 out to this carbon right here, all right, or numbering from all this carbon all the way out to the start of the second double bond, the way that we talk about these fatty acids is to talk about them based on how many carbons in from the very last carbon in the chain is where our double bond starts. So because we count 1, 2, 3, 4, 5, 6, that's the first carbon of this carbon-carbon double bond, we would call this an omega-6 fatty acid. This is also 6, 7, 8, an omega-9 fatty acid because there's another double bond that starts at the 9th carbon in. Have you guys ever heard of omega-6 fats before? What about them? Yeah, they're really heart healthy fats. All right, omega-6 and oatmeal just doesn't have very much fat in it. If you're trying to get fat from eating oatmeal,
I guess I just thought about the heart on the front of the oatmeal.
yeah, lots of food products claim to be heart healthy.
That's one in nutrition that she talked about. It's good.
Well, oatmeal has a good balance of different types of carbohydrates that are not high glycemic index. In other words, like you don't get such a rush of monosaccharides that you spike your blood sugar a lot when you eat oatmeal. At least not when you eat it unsweetened, which most people don't, right?
So when we name these, are we, oh, okay, I just saw that other. Never mind.
Becca, what's your question?
Oh, I was just gonna ask because you said that then there's another double bond at
We
9.
could call this both an omega-6 and an omega-9 fat.
Okay,
Well, omega-6 dash omega-9.
okay, so you would have to like include both of those.
We're
in
not
the-
going to talk about the formal way to name fatty acids, but there is a naming convention even within IUPAC, the formal way of naming, where we can talk about where the double bonds are based on the omega carbon instead of the alpha carbon. All right. Now, if we were naming this based on IUPAC naming, here's how I would name it. All right. Let's count our number of carbons in linoleic acid. Actually, I'll just go back to the last page. All right. Linoleic acid is 18 carbons long. So this is an 18 carbon long fatty acid. All right. Where's our first double bond? Starting from the carbon number 1. 1, 2, 3, 4, 5, 6, 7, 8, 9. All right. And then another one at 10, 11, 12. So if I was naming this based on general naming convention, a 20 carbon long alkane, does anybody know what the prefix for 20 is? Like 10 is deca, so decane would be a 10 carbon long alkane. If you don't worry about it. We've never talked about it, all right? But a 20 carbon long hydrocarbon would be called icosane. Wait, what? What just happened? Did you guys see that?
We've seen it and then it's here.
Yeah, there's a blank screen for us.
I know, it is for us too. Don't worry about it. Not your fault. Oh, it literally died. It canceled itself. It's like, I don't have the will to live. So if we had a 20 carbon long alkane, we would call that icosane. That's how I'd spell it. So tell me what we would call a 20 carbon long carboxylic acid.
Ico, pentanoic acid?
Yes. So pentate will become
Yeah.
pentanoic acid, right? Icosane becomes icosanoic acid. That is a 20 carbon long carboxylic acid. Now, the carboxylic acid that we're looking at has Christmas decorations. It has an alkene at carbon 9 and carbon 12. All right. Do I want to talk about how to name alkenes when they're not the main functional group? No, I'm not going to talk about it.
So
Sorry, and I'm just confused as why we started counting the other way, like instead of the.
So I'm just telling you, I was trying to tell you that we could name this using the IUPAC naming scheme.
Okay. Oh, I see.
Does that
And
make sense? There is a
yes.
way to name this using IUPAC,
And
but
that
we
counts
would
the other.
have to count from carbon 1, and it's a lot longer to get to our double bonds that way.
Yeah, okay, got it.
Biologists and biochemists tend to use this omega way to classify fatty acids because it's easier to count from the very last carbon in the chain because you get to your double bonds faster. Does that make sense?
So does that answer why is it important to get to the double bonds faster?
Well, it's a common way for us to name those.
No, on here though, you asked, it says, how are fatty acids numbered? Which is the omega carbon atom?
Yep.
And why is it important?
Why is the omega carbon important? Because, yeah, the double bonds tend to be closer to the omega carbon than the one at the beginning of the chain. So waxes are one of the types of hydrolyzable esters. All right. Remember, hydrolyzable means we can split them apart into pieces. All right. The two functional groups that we know of to be very hydrolyzable in acidic conditions, esters, amides. We can also talk about hydrolysis of acetals or hemiacetals in acid. So waxes have an ester as their important hydrolyzable functional group. All right, so you can see we can form them by putting together some sort of fatty acid and then some sort of alcohol. The alcohols also tend to have a really long hydrocarbon part when we're talking about waxes. So here is an example of the formation of a wax. This is a wax that is present in sperm whales. All right. So you can see we've got a really long hydrocarbon chain on that fatty acid. And then we have an alcohol that we're adding to it that is also very long and nonpolar. You put those together and essentially this is very shorthanded, right? So this is a condensed structure. We got a really long chain coming out one side of the ester and a really long chain coming out the other side. That basic structure is what waxes look like. So waxes are very, very hydrophobic. That's another word for nonpolar. All right. One of the things that they're used for is the sort of protective nonpolar coating on birds' feathers that allow bird feathers to essentially like bead water off of them very easily, like water off a duck's back. There's real in that. All right. So especially waterfowl of all kinds make tons and tons of these waxes and then groom them onto their feathers. And essentially they've got like the Filson waterproof coat all the time.
For the same question that was regarding the omega carbon, it says which are produced biologically, it was unsaturated fats, right?
Unsaturated and saturated fats are both biologically produced. So particularly when we're talking about unsaturated fats, the natural ones are cis. The double bonds are cis double bonds, not
So that's
trans.
which are produced biologically.
Yes.
Cis, not trans.
Yes.
Oh,
Sorry, one more thing. On the topical videos for this chapter, you had crossed out waxes and said we wouldn't be.
Well, we don't need to focus on those to like understand the functional group. All right. This is a
Oh.
sort of good like lead in to the kinds that we'll focus on more. Now, in terms of their use biologically, waxes have some interesting uses, but they don't tend to be molecules that we make a truckload of because they're not used to do some important job within a cell. All right. So they can be important in terms of like an organism's function. For example, like without waxes, those bird feathers would soak up water and birds would sink, but they're not really important to birds at a cellular level for the function of like a cell at a time. The other types of fats do have really important intracellular roles, and that's why we'll focus more on those. But this is a good lead in because the same type of hydrolyzable functional group, the ester that's present in waxes, is also present in the other types of lipids that do have really important intracellular roles. Becca, is that okay? So we're not going to focus on, that's waxes, all right? I gave
Okay, I was
you
just
a couple of them.
here.
Yeah, they're really not very important compared to the other types of fats, but it doesn't matter for us taking a second to look at them because they have the same sort of hydrolyzable functional group that a lot of the others do, the ester functional group.
Gotcha.
So now you guys have heard of these before, triglycerides. This is one of the things that your doctor checks the levels of in your blood to see how healthy you are. All right. High triglycerides means that somebody is probably storing more fat on their person than they should be. So triglycerides are put together from these parts, all right? A glycerol and 3 fatty acids. Now, this is just sort of a cartoony diagram of that, but I want you to be able to show me what that looks like. So here is a sort of shorthand version of that. So you can see glycerol is a 3-carbon molecule with an alcohol functional group on all 3 carbons. If we put that together with 3 fatty acids, and we should say 12 to 20 carbons, not 11 to 19, because they generally have, oh, the R groups have 11 to 19. Yeah, I suppose. So I would have written this a little bit differently. I would have said the fatty acids have 12 to 20 carbons. Now that means that the R group, which is 1 less than the entire fatty acid, would have 11 to 19. Sorry about that. So 3 long fatty acids, combine that with glycerol. And what type of thing do we make when we put an alcohol and a carboxylic acid together? We get an ester, all right? So triglycerides or triacylglycerols have 3 ester functional groups in them. You don't have to understand the difference between simple and mixed ones. In biological systems, there is a gnarly mixture of all sorts of different fatty acids when we make triglycerides. Your body is circulating so many different fatty acids around, and the mechanisms for formation of these triglycerides, they're not particularly picky about which fatty acid that they put in. So in biological systems, we normally get a mixture of lots and lots of different fatty acids in our triglycerides. So I don't care if you understand the difference between simple and mixed ones because they're both made and they do what they're supposed to regardless of which of the 3 fats there are there. So lots of animal fats end up containing saturated fatty acid side chains, all right? Remember that we said that saturated fats don't have carbon-carbon double bonds in them, and that saturated fats tend to melt at higher temperatures than unsaturated fats. So lots and lots of animal fats are saturated triglycerides like this. They're solids at room temperature. So triglycerides tend to be the way that we store lots and lots of extra energy as fat. All right. And unsaturated triglycerides contain at least one unsaturated fat and vegetable oils. All right, those are the basic structure of most vegetable oils. You would think that if saturated fats are unhealthy and unsaturated ones are healthier, that more double bonds might be better. That's not necessarily the case. All right, there are some specific health benefits of monounsaturated fats that polyunsaturated fats don't have. But generally speaking, avoiding saturated fats tends to be a good idea. All right, it's impossible to do it completely. Even the healthiest fats, like olive oil, for example, has some saturated fat in it. All right, it's just the proportions. So I do want you to understand the difference between what a monounsaturated fat means and what a polyunsaturated fat is. It just has to do with how many double bonds. All right. So here is an unsaturated triglyceride. Is this a monounsaturated triglyceride or a polyunsaturated triglyceride? Poly? How many double bonds are there in there? How many carbon-carbon double bonds?
One.
In the chain.
In any of the chains.
Yeah.
All right. There's only one. So is it a polyunsaturated or it's mono? Yeah. It is a monounsaturated triglyceride. So the difference between whether we call something an oil or a fat is more of a stylistic thing than anything else. All right. But we tend to use the name fat for something that is a solid at room temperature and oil for something that is a liquid at room temperature. So why don't we call coconut oil coconut fat? Probably because coconut oil sounds cooler, right? Yeah, coconut fat doesn't sound nearly as appealing as coconut oil does. But coconut oil is a solid at room temperature. Is it a solid at room temperature always? No, on a hot day, it can be a liquid. So calling it coconut oil, I suppose, is okay in Australia, but not in Canada. I don't know. All right. Yeah. So fat and oil, those terms tend to be reserved for what state of matter the thing is at room temperature, but there's some play there. So you'll catch me calling things oils that tend to be solids at room temperatures, and you'll catch me calling things that tend to be liquids at room temperatures fats sometimes. They're sort of interchangeable terms, but we tend to use fat when we're talking about something that's a solid at room temperature. We tend to use oil when we're talking about something that's a liquid at room temperature. You might, it might make perfect sense to you to realize that since we tend to use the word fat to talk about things that are solids at room temperatures, that means that those kinds of triglycerides tend to have very few double bonds. Okay, so does everybody understand the difference between chemical structure between things that we call fats and things that we call oils? So since oils tend to be liquids at room temperature, we know that means that those things tend to have more carbon-carbon double bonds in their structures. All right. So take a look. I really like this table a lot. It's instructive as heck. All right, take a look at olive oil and its proportion of different types of fats. We think of olive oil as like the model healthy oil, right? 72% of the fatty acids in olive oil are monounsaturated fats. Only 13% are saturated. Do we think of coconut oil as a healthy oil? Some people do that don't read very much about it, but 87% of the fat in coconut oil is saturated, all right? Compare that to lard. Actually, lard gets a bad rap, but 44% of the fatty acids in lard are monounsaturated, the same healthy kinds of fat that are in olive oil. You could see that lard is definitely a healthier choice for a cooking fat than butter is. A lot less saturated fat. All right. So that's one of those things that's sort of changed in the traditional American diet over the last 50 years. We use butter to cook a lot more things and lard a lot less to cook things. Guess what? We probably shouldn't have switched. For one, lard is more delicious than butter could ever be as a cooking fat. All right. Unless you're vegan, in which case, like, use a vegetable oil, and that's fine. We got nothing against vegans at all.
What would fall consideration of lard?
What you buy lard, it's like animal fat. Yeah. So you used to, I mean, you can still go to the butcher shop at any grocery store and buy some form of lard. It could be
Is it-
extra pork fat to use or extra, it's normally like cow fat.
Oh, it is? I didn't know that.
Yep. So lards tend to be,
Like.
it's
cleaned
white.
and-
Yeah. Yeah. They render it, which
'Cause
means
you
that
can
they
make
heat it
your
up.
own.
You can make your own, sure.
That's why I'm asking, because I think I've seen like, TikTok and everything these days, but like.
Yeah, the way you make your own normally is to like take a really fatty cut of meat and heat it until all of the fat melts and gets to the top. And then you skim the liquid fat off the top and then strain it and then let it re-solidify.
Yeah, okay.
Yeah,
she
so
took,
that's
like,
how
a
you
bunch
make
of
your-
ground beef and then poured it off,
Mhm.
and then just like heated it in water and then put it in cold water until it cooled and cleaned it enough that it was. I just wasn't sure
Another
if it was
common
something-
thing that, I mean, I suppose it could be considered lard, but not as commonly. Like, if you cook bacon,
wasn't
using the
actually-
bacon grease to cook things in, it's probably a healthier choice than using butter. So fats have lots and lots of different jobs in the body. We use them to make cell membranes. We use them as insulation. And then they're also a very efficient way to store excess energy for later use. You're not going to be tested over dietary recommendations, but unless there's a specific reason. Otherwise, like for example, some people following the keto diet eat like 2/3 of their calories from fat and like 30% from protein, and then try and keep carbs out of their diet as much as they can. All right, not very delicious, but it does trigger some physiological adaptations in the body that make it easier to lose weight. Also, if you're not eating any carbohydrates, you probably lose some of the will to live, which makes it easier to calorie restrict, right?
How would each be oriented when present in biological membranes? Is
We
that-
haven't gotten to that yet.
Okay, I'm just checking.
Quit asking all the questions on the, I only want to write down what's in the exam
No, I
review
just wanna make
guide.
sure I didn't miss
We haven't
it.
gotten to it yet. We haven't
That's
gotten to it
all.
yet. We haven't even talked about phospholipids or the phospholipid bilayer. All right, we'll get there.
But we talked about the triglycerides, so I wasn't sure. Oh,
So if you are wondering about what sorts of foods you should avoid if you want to avoid saturated fats, red meats, cheeses, butter, fried foods, like all the delicious stuff has lots of saturated fat in it. Bummer. And there are lots of delicious things in here as well. Avocado is a good mix of healthy fats and also tastes very fatty and delicious. So I love avocado. If you have cats love avocados too, by the way. So if you want to give your cats a treat and not give them like a little slice of raw meat or something, avocado, cats love that stuff.
I assume you've tested this.
Oh yeah. Every cat I've ever had has been bonkers for avocado slices. They go crazy. So where do we get trans fats from? Trans fats tend to be artificial. All right. So if you look at any heavily processed foods that have a lot of fat in them, they tend to have those trans double bonds in them. All right. They have lots and lots of there are lots and lots of problems metabolically with us processing these, and they have pretty pronounced health effects. So we want to stay away from trans fats as much as we possibly can. If you take one practical thing away from the chapter on lipids, stay away from trans fats. All right, they're bad. So unsaturated fats actually have like the exact opposite effect that saturated and trans fats do. They actually allow us to decrease circulating blood cholesterol. That's not something you're going to be tested over, but unsaturated fats are really healthy for a host of reasons. So that's one of them. All right. And omega-3 and omega-6 fats particularly are really helpful at lowering cholesterol, which leads to us not having as big of a risk of heart attack, because heart attacks, unless you have some sort of genetic condition that your heart like isn't formed correctly, the main reason that people have heart attacks is what happens to them? Clogged arteries. What are the arteries clogged with?
Fats.
What kind of fats?
Saturated.
Cholesterol.
Cholesterol.
Cholesterol. So cholesterol is what deposits on the inside of our arteries and can lead to clogged arteries and heart attacks, right? So things that we can do to decrease our circulating cholesterol in our blood are things that are protective against heart attacks. One of the main things that we can do to protect ourselves against heart attacks is to eat more omega-3 and omega-6 monounsaturated fats. The reason that that happens is through a host of biological processes. All right, so to understand all of metabolism, that's more than I actually really understand completely. I'm a molecular biochemist, not a systems biochemist. So I think about the smallest pieces instead of the whole system at a time. People that understand metabolism can talk to you about all of these different mechanisms by which these omega-3 and omega-6 fats lower our amount of circulating cholesterol in our blood. So doing that is a direct prevention against heart attacks. It's pretty awesome. All right. And there are lots of things on the list for omega-3 and omega-6 fats that are really delicious things. Salmon, for example, has lots and lots of heart healthy fats. So everybody, especially in the Pacific Northwest, unless you're a vegan, you should be eating salmon once a week out here.
If it's like
But never farm salmon.
Farm salmon still has those omega-3 and omega-6 fats though.
Yeah, but they dye that flesh and they swim around in their own stuff and they don't swim far, so it's not as good.
They still have lots and lots of those fats. Now, I'm not saying that we would prefer farm salmon, but would you expect a farm salmon or a wild salmon to have more fat in it?
A farm salmon.
The farm salmon, right? Because the
Ugh.
farm salmon are not swimming long distances all the time. Okay, so like in terms of like pure clinical benefit, I have not researched this, so it's not a statement of fact, but my inclination in my head is that farm salmon would probably be healthier to eat even than wild salmon because they would have more of the omega-3 and omega-6 fats. That is not a statement of fact. I have no evidence to back that up, but just thinking about their lifestyles, I would anticipate that farm salmon have even higher levels of omega-3 and omega-6 fats. That could be hogwash, but I don't understand the mechanism how that could not be true. So
too, so it may not be.
yep, it may not be
Exactly what I
true.
was gonna
It could
ask.
be a difference in quality of food, making different fats. But generally speaking, creatures that are sedentary make more fat. So I would anticipate more fat on farm salmon. And if they're making the same kinds of fat, then the farm salmon would probably be even healthier for your heart. All right. Eating salmon, whether it's farmed or not, is better for your heart than not eating salmon. So making the choice to switch from a steak to salmon, if you switch half of your steaks to salmons, you'll probably live longer as a result of that. That's true. So trans fats, you'll definitely get tested over that. They're lame. All right. Any beneficial effects from carbon-carbon double bonds, they don't work if they're trans double bonds. Your body knows what to do with cis double bonds. We have enzymes for that. Your body freaks out at trans fats because our enzymes don't recognize them the same way. All right, so trans fats are artificial, they're not natural, and they tend to increase cholesterol levels in the blood. They do the exact opposite of what those natural unsaturated fats do. we've talked about ester hydrolysis before. So just like hydrolysis can happen of esters using acid, enzymes can do that hydrolysis as well. So this tends to be the very first step in metabolism of triglycerides. Like, for example, if you're at a caloric deficit and you need more energy, your body starts burning fat as energy, right? So it starts to break these triglycerides into pieces. The first step in that metabolism to get the energy back that's stored in there is hydrolysis of the triglycerides. We break our triglyceride into a glycerol and 3 of those fatty acids, whatever they are. So triglycerides tend to be our storage form for excess energy. All right, we store fat in lots of places on our body. And those adipose cells, the fat cells, are the ones that are sort of the place where all of that excess triglyceride goes. It turns out that the number of those cells is constant. So whether you are carrying lots of extra fat or not is essentially like how big those individual cells are based on how swollen they are by storing more or less of those triglycerides. That's really interesting. I've read that there are some interesting things that happen to people that have liposuction, because that actually does change the number of adipose cells in certain tissues. So people that have liposuction tend to not gain as much fat in those places where they had liposuction, even if they gain weight other places, because the number of fat cells in the body tends to remain more or less constant. So if you don't have as many fat cells in your midsection because you had liposuction, you're probably never going to gain as much fat around that midsection as you did before. All right. You could think of that as a good thing, but you could also look grossly misshapen if you gain lots of fat cells other places and you end up looking like much bigger in one place than another. So an interesting set of thoughts there.
There's no way to get rid of those cells.
Liposuction. You're literally vacuuming out some of those cells, right? In terms of fat loss, like losing weight, you don't lose fat cells.
I don't know why
You
it's-
just
it's
lose
not
volume.
happening.
Yeah, you just lose volume in those fat cells, not fat cells themselves. I think that's really interesting. So even a bodybuilder at 6% body fat, they haven't lost fat cells. The fat cells are just very, very teeny. The enzymes that we use to hydrolyze our triglycerides are called lipases. So the name lipid is even in there. And any enzyme that has an ACE as its suffix tends to be something that does some sort of a hydrolysis reaction. All right, so lipases hydrolyze lipids. Peptidases. What do you suppose a peptidase hydrolyzes? A peptide. All right. So if we completely metabolize triglycerides, we end up making CO2, which we exhale. We make water, which is no big deal because there's already some of it in our body. And there's also a great deal of energy that's produced there. In other words, in a biological system, energy means that we convert the energy that is stored in those molecules into the synthesis of a lot of ATP, a cellular energy currency. So here's kind of a cool microscopic picture of adipose cells. The only difference between what that would look like in a person that was carrying a lot of extra fat and one who wasn't is the size of those individual cells. All right, they would be much more swollen or much less swollen, depending on how much triglyceride was currently being stored in them. You've probably, if you've had biology classes before, heard about the amount of energy contained in a gram of each of those types of like fats and carbohydrates and proteins. So it turns out that fats contain almost twice the amount of energy per gram as the other sources of stuff in your food. So you can only eat half as much mass of fat if you want to eat the same number of calories. All right, 9 calories per gram. So proteins and carbohydrates contain about 5 calories per gram instead. This is old news. I think lab 2, we made our soaps. All right. So soaps are what we get when we have fatty acids that we have treated with some sort of a base. So remember that when we treat a carboxylic acid with a base, in basic conditions, carboxylic acids lose their hydrogen and end up with a negative charge on that oxygen. When we do that to a fatty acid, it becomes a soap. So here is a sort of picture of what it would look like if we made soap out of this particular triglyceride. So notice that in that soap at the end, we end up with glycerol, which can be washed away in the aqueous part. And then we end up with this very long carboxylate. Remember, we call carboxylic acids without their protons carboxylates. So that's the structure of a soap molecule. We also talked about how soaps work all the way back in lab 2. All right. So soaps work because the nonpolar part of the soap, the hydrocarbon part, is really good at dissolving things that are nonpolar, like greases and oil. The polar part of the soap can dissolve well in water. And I don't have a really good picture of that, but here's how it actually looks three-dimensionally speaking. If we draw a cartoon version of a soap molecule, where the circle is our polar head group, and the hydrocarbon part is represented by that sort of squiggly tail. The way that those things actually look when they organize by themselves in aqueous solution looks something like this. I've probably drawn too many of these just to save time. I'm going to put gaps where there actually really aren't gaps. So in a situation like this, the entire outside of this three-dimensional thing is polar head groups. I'm drawing it two-dimensionally, but think of it as a sphere. All right, the outside of the sphere is interacting with water. The inside of the sphere is interacting only with other nonpolar tails. So the entire inside of this sphere is very nonpolar. And draw that in a different color so we can actually read it. So greasy dirt can dissolve very nicely. Oils and grease can dissolve very nicely on the inside of these structures that are created from soap molecules in solution. Does anybody remember what we call these things? This three-dimensional structure that's
Micelles.
yeah, this is called a micelle. So soap molecules, pure fatty acids that are treated with bases to make soap, they form these three-dimensional structures all by themselves in solution called micelles. All right. The micelles dissolve very nicely in water, but the inside of the micelle is nonpolar, so it can dissolve other nonpolar stuff like grease and greasy kinds of oily dirt. That is the way that soaps can clean clothes or clean hands. So if you ever get oil on your hands and want to clean them off, you wash your hands with some soap and the oil is magically gone. It's because the oil dissolved inside of those micelles. Pretty awesome. So this is what fatty acids look like when they self-organize, when they form a three-dimensional structure in aqueous solution. The other types of lipids that we're going to talk about organize differently than this. All right, so it's really important for us to understand the difference between what fatty acids do when we put them into solution, especially when they're deprotonated at the end. They form these micelles. And what happens when other kinds of lipids go into aqueous solution. So other kinds of lipids will also organize into structures, but the structures are different. So the first type, oh no, there we go. The first type of lipids that we'll talk about other than the triglycerides is a kind of lipid called a phospholipid. Now, phospholipids have a couple of different types of structures, all right? But if we look at if we look at a model of either one of these kinds of phospholipids, the actual shape of these phospholipids is always very, very similar to one another. So the kind of phospholipid I want you to remember the most about is one that looks like this, where we have a glycerol with 2 fatty acids and then a phosphate and an alcohol on our third position. Note that phospholipids look a lot like triglycerides. What is a triglyceride? A triglyceride is a glycerol with 3 fatty acids, right? Phospholipids are almost the same thing, except one of our fatty acids is replaced. There's also another kind of fatty acid where instead of having a glycerol as its backbone, we have this other kind of molecule called sphingosine. Now, sphingosine has its own fatty chain on it, which means that putting just one fatty acid on a sphingosine gives us 2 long fatty chains and then a phosphate and an alcohol. So in terms of their actual shape and job, the glycerol-based phospholipids and the sphingosine-based phospholipids, they work the exact same way because it's 2 fatty chains and then a phosphate and an alcohol. Whether you're talking about those fatty chains coming from fatty acids or a fatty acid and a sphingosine, it's 2 long hydrocarbon chains and a phosphate. All right, that's what constitutes what a phospholipid looks like. You do not have to remember the structures of those alcohol functional groups. There are a number of different ones that can be attached here, all right? The most common one is something called ethanolamine. So here is an example. Again, we're not drawing out the entire fatty side chains because it takes too long, but they're there, all right? So here is another example, choline of one of the alcohols. And choline happens to have a nitrogen with 4 bonds to carbon in it. That's kind of an interesting biological thing. Again, you need to know that there's an alcohol here. You don't have to know anything about the structure of it. All right. So I do want you to be able to tell me what the parts of a phospholipid are. So a glycerol, 2 fatty acids, and then a phosphate with an alcohol on the other side. So those are the components that we want to remember when we're talking about the most common phospholipids. So because phospholipids have a polar head group, so the polar head group on a phospholipid is the part with the glycerol and the phosphate and the alcohol on it, all right? That's the polar head part of a phospholipid. Those 2 fatty acids that are attached form 2 tails. So we often depict phospholipids as a polar head group that has 2 nonpolar tails on it. The important thing here is that lipids with 2 nonpolar tails, they can't organize into micelles anymore. All right, so I want you to think about why that might be. Why do you suppose that a lipid with 2 nonpolar tails might not be able to make a micelle anymore?
Because they're more hydrophilic than hydrophobic, or the other way around?
Well, they're definitely more hydrophobic because there's more nonpolar part to them, all right? But it turns out that it's an even simpler explanation than their relative polarities. It has to do with the amount of space on the inside of the micelle. Imagine trying to cram twice the nonpolar tails into that space. There just isn't enough space to make that kind of structure work. It's a good thing too, because phospholipids, if they made micelles instead of what they actually make, life on our planet wouldn't exist in the form that it does. All right, so phospholipids self-organize into these structures. And again, this is a 3-dimensional thing represented 2-dimensionally. They organize into these sheets that have polar head groups on one side and on the other side, and then the nonpolar tails pointing in toward the middle. We call this structure a phospholipid bilayer. Anybody know what phospholipid bilayers are used for?
Cell membranes.
Cell membranes, all right? Pretty much every kind of biological membrane you can think of is a phospholipid bilayer. Now, it's not just phospholipids that are in those cell membranes. There are proteins sticking out of the membranes, and there are pieces of cholesterol in there, all right? But phospholipids are the main component in all cell membranes. So if phospholipids did not self-organize into these bilayers, we couldn't have cells. That seems pretty important, right? So here is a sort of model picture of what a phospholipid might look like. Let's identify the parts in there, all right? Right here is our 3-carbon glycerol. Here are our 2 ester functional groups to those fatty acid side chains. And then here is our phosphate and the alcohol that's attached to the phosphate. All right, the alcohol is actually attached here at that oxygen atom, and we call it a phosphoester bond. So you can see that that polar head group, because of the alcohol, has a couple of different charged atoms. That's a very polar head group, and then our 2 nonpolar tails. Now, I told you that not all phospholipids are derived from glycerol. Some of them have sphingosine instead. So the examples of those, they're called sphingomyelins, all right? But if we take a look at a 3-dimensional picture of those, here's what sphingosine looks like. It has its own nice long hydrocarbon tail on it. So when we put sphingosine into a phospholipid, we still end up with 2 long nonpolar tails and then a phosphate part, all right? I thought I had a model picture of that, but I guess I don't. I just have a picture. All right. In this condensed structure, we can see the sphingosine part of our sphingomyelin. We can see a long fatty acid part. Note that the fatty acid in sphingomyelin is a little different because it's attached through an amide bond instead of an ester bond. All right. Not a big difference, because when we think of phospholipids, essentially we're thinking of a polar head group that has phosphate in it and 2 nonpolar tails. That's always what we think about when we think about a phospholipid. Polar head group with phosphate and 2 nonpolar tails. It doesn't matter whether it's derived from sphingosine or whether it's derived from glycerol. They do the same things. So it turns out that sphingomyelin or other kinds of phospholipids derived from sphingosine, they happen to be present more in the cell membranes of nerve cells, all right? Because nerve cells have lots of jobs that involve conduction of signals, those membranes that have sphingomyelin in them tend to be membranes that the components can move around quicker. We don't have to understand nerve conduction for this class, but here's a picture of those sheaths of myelin that surround. Myelin are kind of cells that are that have lots and lots of sphingomyelins in their phospholipid bilayers. We don't have to understand everything about cells in this class. This is not a full biology class, all right? But I do want you to remember that phospholipids are used to form cell membranes. That's an important factoid. All right. So because phospholipids have a nonpolar part in the middle in the bilayer, all right? So in that phospholipid bilayer, there's a big nonpolar part. That means that any molecules that want to get into a cell have to pass both a really polar head group part and a really nonpolar piece in the middle. So that means most larger molecules that are polar cannot get through the cell membrane by themselves. All right. It is a way of cells controlling what gets in and what gets out. What's really interesting, at least to me, is that the phospholipid bilayer, the cell membrane, actually lets water in and out pretty easily. So I think a lot of it has to do with water just being small enough to sort of squeeze past. But most other larger molecules that are polar, they can't do that. The other thing that can't get through the phospholipid bilayer is dissolved ions, like sodium ions or potassium ions. That's really important because there are ionic gradients in the cell that allow us to harvest energy. One of those ionic gradients is a difference in the concentration of H+ ions. So if you've had biology classes, you'll know that a proton gradient is what's used to make ATP in the cell. All right. If cell membranes just let H+'s go past, we'd have no way to make ATP. So the fact that cell membranes resist the passage of ions, that's really, really important. So because the cell membrane allows some stuff to go in and out, namely water, all right, we call the cell membrane selectively permeable. It lets some stuff in, but doesn't let other stuff in. It lets some stuff out, but doesn't let other stuff out. So how do we let other stuff out or let other stuff in if the cell membrane doesn't allow it? Does anybody know?
Don't we encapsulate it in the cells and then?
You can, yep. So endocytosis and exocytosis are definitely mechanisms that we can use. But we also have all sorts of other kinds of pumps and selective ion channels and things. So there are proteins whose jobs are to regulate passage of ions into and out of the cell. All right. There are other kinds of proteins whose jobs it is to selectively let certain molecules into and out of cells based on size. Like we call those porins. All right. Essentially, like they're a controlled pore that allow passage of certain things into and out of the cell. So we've already talked about this. Phospholipids are the major component of cell membranes, and they have those two hydrophobic tails. And they make a lipid bilayer with the polar heads on the outside and the non-polar tails are on the inside. So here is a much better picture of that three-dimensionally than I could make. One of the things that I find really amazing about the phospholipid bilayer is that even though it looks like it's perfectly packed, it's really not. So depending on what these two nonpolar tails look like, one of those tails might have a cis double bond in it someplace, which would mean that it's kind of crooked, right? It doesn't go in a straight chain. It's kinked. So that means that those phospholipids that have unsaturated fatty acids in them, they don't pack as neatly as the ones that have saturated fatty acids in them. That's one of the ways that we can regulate how mobile those components of the membrane are. All those components are actually moving around in the membrane all the time. All right, we actually have a name for that model. We call it the fluid mosaic model of membranes. You don't have to remember that name yet. Not that big of a deal. All right. But fluid mosaic, does anybody want to know what a mosaic is? It's like an art where we make things from like macaroni paintings, right? Where you glue all the little pieces of macaroni to make a portrait. That's a mosaic. A lot of little pieces. So a fluid mosaic is a lot of little pieces that are all moving around. That's what membranes look like. The individual pieces are not stuck. They're moving. It's awesome. Here is a better picture of what membranes look like. It ain't just phospholipids. We also have cholesterol. We also have carbohydrates stuck to the surface of some cell proteins. And then we have some proteins that are going all the way through the membrane and some that are stuck on one side or the other of the membrane. So cell membranes are really complex. And not all cell membranes are created equally. All right. So for example, there are cell membranes on the outside of cells. There are also cell membranes that surround an organelle in the cell. Like for example, mitochondria have their own membranes. So mitochondrial cell membranes, even though they're phospholipid bilayers, just like the outer cells, the stuff in those is different. All right. Every membrane within the cell that has its own job is some version of phospholipid with other stuff in it. All right, Brittany, how far are we in our study guide?
I still didn't see, or maybe I just didn't add it with it. How would, each be oriented when present in biological membranes?
How would each what?
Each of the, like, triglycerides, phospholipids, glycolipids.
So triglycerides are not part of biological membranes.
Right.
That's how they're involved. All right. Phospholipids, we know how they're oriented, right? So in the bilayer, their polar head groups are pointing toward the outside and the non-polar tails are pointed toward the inside of the bilayer. And then for fatty acids, we talked about the micelle, the structure of the micelle, where the polar head groups are on the outside interacting with the water.
Is that the glycolipids and cholesterol?
Glycolipids are very similar to phospholipids. So the only difference, we haven't gotten to glycolipids yet.
Okay.
We'll get there next.
And
All right.
cholesterol?
And cholesterol. Yeah, we haven't gotten to either of those yet.
And then the next question revolving around that is, except triglycerols, what are not in biological membranes? Since they're
Waxes.
polar.
Waxes are not in biological membranes anyplace.
How are lipids and archaea different?
We'll talk about it. All right, we're not just going to cover the study guide materials. We have to talk through the chapter so you guys get an appreciation for these different kinds of lipids. So answer those questions as they come up. We'll get to them. We'll get to it. We don't need to talk about transport across membranes. If you're interested in how membranes regulate what gets in and what gets out, Brian Brady does an amazing job of talking about that in his biology class. All right, so we talk about facilitated transport and we talk about transport with gradients. There are lots of different mechanisms for how the cell gets things in and out. All right, it's amazing and really complex. So this will be like more than one entire test probably in the biology class, if I was guessing. So we have not yet gotten to a couple of types of lipids. Cholesterol is the first one. Cholesterol is really different because it doesn't have an ester functional group anywhere in it. All right. So cholesterol is a type of steroid. Steroids tend to have this system where there are a bunch of fused hydrocarbon rings. So it's really easy to see when we're talking about some sort of a steroid because they all have that sort of fused ring system in them. You don't have to know how to number the steroid skeleton. I actually remembered that at some point in grad school and then forgot it. All right. But here is the most important of the steroids, cholesterol. So cholesterol does have a polar head group. Where's the polar head group in cholesterol? The top? Right here?
Oh,
Is
where
that
that
polar?
alcohol is, or phenol.
Yeah, that alcohol functional group. That's the only polar part of the entire cholesterol molecule. All right. If you said that was the top, then I challenge your directional sense a little bit. All right. But I think we all know that the hydroxyl group there is the only polar part of that entire molecule. So if we were thinking about how cholesterol might integrate in a biological membrane. So for example, let's say we have a cholesterol in our phospholipid bilayer someplace. All right. What do you suppose would be pointed out with the phosphates? And what do you suppose would be pointed in with the nonpolar tails? The hydroxyl group of cholesterol would be pointed out toward the outside of the bilayer. And the rest of the cholesterol will be pointed inside along with those nonpolar tails of the phospholipids.
That's what all those questions are asked.
Yeah. So control of the amount of cholesterol in the bloodstream is really important because if you have too much circulating, it can deposit on the inside of arteries and close them off. That's bad. Cholesterol is not circulating in your blood all by itself. All right. it is transported using proteins because otherwise it would be insoluble and you would get just deposits of cholesterol. It would precipitate in your arteries. That'd be really bad. Instead, we have these class of proteins called lipoproteins, fat transport proteins. All right. They hold lots of phospholipids on their surface, and they have a protein core underneath them. So there are a couple of different types of lipoproteins that transport cholesterol. You don't have to understand all of the roles for these, but it turns out that lipoproteins that are taking only small numbers of cholesterol around, those are an indicator that there's just too much cholesterol. And high density lipoproteins are proteins that indicate that you have a healthy cholesterol balance. So when you get your cholesterol measured, what they're measuring is what's the concentration of LDLs in your blood and what's the concentration of HDLs in your blood. So if your LDL concentration is too high or your HDL concentration is too low, either combination of those could indicate that you are not processing cholesterol very well. You have too much circulating cholesterol that's unhealthy in your blood. So this is essentially what a lipoprotein looks like. All right. The protein piece up here, and then a phospholipid monolayer that is sort of covering a big core of cholesterols. This is the way we can carry around cholesterol, which by itself would not dissolve in water very well at all, not polar enough. But if we can hide it from the water on the inside of a phospholipid core, we can transport it. So LDLs sometimes drop cholesterol off on the walls of arteries. That's bad. HDLs are an indicator that there isn't very much extra cholesterol around. So their presence is telling us that the LDL should probably be at a healthy level as well. So having too high a concentration of LDLs in your blood, that's what we call bad cholesterol. All right. Note that cholesterol is more of an indicator of overall metabolic health than actually the amount of cholesterol that you consume in your diet. So for a long time, dietitians recommended not eating very much cholesterol in your diet because they thought that eating that cholesterol was the mechanism by which bad cholesterol was increased in your blood. It turns out that your body is really, really good at making cholesterol. So no matter how much of it you eat, your body makes many times that amount all by itself every day. So dietary cholesterol is probably not very important in terms of whether or not your body has a healthy cholesterol balance. Instead, it has to do with the overall quality of the diet that you eat. All right. So if you're storing lots and lots of triglycerides, the other underlying metabolic things mean that you're probably producing more cholesterol than you should be at the same time. It doesn't have to do with dietary cholesterol. All right. But cholesterol gets made too much when you're making too much of other kinds of fats as well. Pretty neat. All right. The higher your HDL within reason, the better. The lower your your LDL within reason, the better. So you should know which is the good kind of cholesterol and which is the bad kind. Here is the difference between what a healthy artery looks like and what one looks like when it has had lots and lots of cholesterol deposit on it. That's not very much space for blood to flow. All right. The other thing that does is if you're narrowing your blood vessels, what happens when your heart is pumping blood through that area? Do you think that there would be more or less pressure pushing blood through that compared to that? Probably a lot higher pressure here, right? So people that have lots and lots of cholesterol deposits in their arteries, they tend to have really high blood pressure too. We're not going to talk too much about statins, but these are drugs that decrease the amount of circulating LDLs in the bloodstream through a number of different mechanisms. All right. Really interesting. They're also some of the most lucrative drugs that exist. Lots and lots of people have high cholesterol in their blood and need to lower it. All right. Because they're not eating a healthy enough diet, or there are genetic predispositions for that as well. All right. Not everything is a diet thing. But statins are multi-billion-dollar a year moneymakers for all the pharmaceutical companies. Some of them have more recently gone off patent, which means that their costs have gone way, way down, which is good. So statins tend to block the synthesis of cholesterol. The exact stage at which it's blocked depends on which statin you're talking about. All right, they don't all work exactly the same way. So if we're talking about other kinds of lipids, we're not going to spend hardly any time talking about other steroids, but steroids are all lipids. Lipids are just any nonpolar biological molecule. So when you hear the word steroid, what do you think about?
Beefing up.
Like anabolic steroids, right? Steroids that that trigger our body to make more muscle. Those are lipids as well. They're just lipids that have very specific biological roles. There are other steroids that are catabolic steroids. They're also lipids, but their particular structure signals our body to break down muscle instead of building muscle. So natural steroids, there's a balance between those. All right. There are also other sorts of steroids that are related to biological sex. So you guys have probably heard of the estrogens. All right. One of the main reasons that women are predisposed to breast cancer is a dysfunction of the signaling process when breast tissue is developing. All right. So there are certain sorts of genetic anomalies that can happen in breast tissue that make that tissue very, very susceptible to having rampant uncontrolled cell dividing, i.e. cancer. Very recently, we have figured out that because those genetic conditions also mean that certain proteins are expressed in a much higher level in that breast tissue, that's become a specific way that we can target those cells with anti-cancer drugs. So if you or somebody in your family ends up with breast cancer at some point now, you have a much, much better chance now than you did even 20 years ago of being completely cured from that cancer, because we have targeted therapies based on the changes in those cells. And those changes have to do with how those cells process certain estrogens. So the main one of the estrogens that's present in, well, it's in both men and women to some extent, but that regulates biological sex for women, estradiol. All right. This is another steroid that's present, generally speaking, in pregnant women. Also, there are some genetic abnormalities in testicular cells that are becoming cancerous. So there are anecdotal evidences that if a guy pees on a pregnancy test strip that's testing for the presence of progesterone and tests positive for being pregnant, there's a good chance that guy probably has testicular cancer. Isn't that interesting?
I've heard that.
Yeah, it's cool. I mean, not cool that somebody would have cancer, but cool that it could be detected by that kind of test. So here are a couple of the male steroids that regulate our characteristics. And these are not exclusively male or female hormones. They're just expressed at different levels in men compared to women. Women make testosterone also. There's a natural range of testosterone production in women. It tends to be lower than the natural range of testosterone production in men. We don't have to talk about anabolic steroids, but here are some examples of those. Notice that all of them have that same sort of fused ring look to them. All right, you can tell what a steroid looks like. You don't have to know any of these structures. You should be able to recognize cholesterol. All right. The important thing about cholesterol is that down here, there's an OH group in it. All right. That OH group is the one that tells you it's probably cholesterol you're looking at there. All right. This is so many things. I thought I had some slides about glycolipids in here. Did we skip them somehow?
Okay, cross that off the list. I don't know.
No. All right. So a glycolipid, we don't need the slide to talk about it, at least. It's easy. So glyco, what do you suppose glyco means?
Cholesterol one. Glycogen, glyco something.
What is glycogen? It's a polymer of, oh, maybe you haven't studied it yet. All right. Glycogen's a polymer of glucose. So glycolipids are lipids that have a sugar as their polar head group. So if we're thinking about the general structure of a glycolipid, they're also derived from glycerol. All right. They also, like phospholipids, have two fatty acid chains on them. The only difference is instead of having a phosphate and an alcohol on the third place on our glycerol, they have some sort of a monosaccharide as their polar head group. Generally, it's a glucose, but it could be any one of a number of things. So if you think about a polar head group and two nonpolar tails, what do you automatically think about? A phospholipid, right? Glycolipids sort of serve similar kinds of purposes. It's just the polar head group is a little bit different. That wasn't so bad, was it? I can't believe that we didn't have a glycolipid slide in there. Lame sauce. So let's take a look, because I think that we've probably answered all of our questions for the exam 3. Are you not going to let
About
me?
the archaea? I mean,
Oh, yeah, we haven't talked about that yet. I'm so excited, and I would have missed it otherwise. All right. So there isn't a slide for this either, but it's amazing. It's amazing. So this is one of the coolest adaptations that living creatures on planet Earth have made to adapting to extreme environments. And it has directly to do with the structure of their lipids. All right. So let me remind you what a phospholipid in humans or bacteria or pretty much any other thing that is not weird as heck. This is what their phospholipids look like. Remember, we have, oh, I don't want to draw that like that. We have our glycerol part. We've got our two fatty acid tails, and we have a phosphate with an alcohol attached on the other side. That's a phospholipid for anything reasonable on planet Earth. All right. Notice that those two fatty tails are connected by esters. So what do esters do? They hydrolyze. If there's acid around, or if there are enzymes around, or if the temperature even gets too hot, they hydrolyze. Now let's take a look at the structure of phospholipids in archaea. Does anybody know what archaea are or where they tend to show up? If you've had a microbiology class ever, you've probably heard of them. If you hadn't, I wouldn't be surprised. All right. they are really, really ancient in terms of their phylogenetic divergence from other creatures. In other words, like really early on in evolution, bacteria and archaea split from one another. So their biological way of doing things is really different. And they have adapted so that they tend to take advantage of physiological places like microenvironments on Earth that other organisms can't live in very well. One of those places where archaea have decided they want to live because nobody else wants to live there is really close to mid-ocean vents, like steam vents at the bottom of the ocean. These archaea are living where the average temperature is like 60 or 70 degrees Celsius. Do you know how hot 60 or 70 degrees Celsius is? Hot. Boiling water is 100 degrees Celsius. Room temperature, 20 degrees Celsius. All right? So 60 degrees Celsius water is hot enough that you could stick your finger into it for a second, but any longer than that, it would be burning. These creatures live at that temperature all the time. That temperature would be hot enough where regular human phospholipids, those ester linkages would hydrolyze. We would cut our fatty acids and have no phospholipid bilayer anymore at that temperature for normal creatures. So archaea must have phospholipids because they have cell membranes. Their cell membranes are made up of phospholipids that do it differently. The structure is different. Phospholipids in archaea are still made from glycerol. But those long fatty side chains are attached differently. What kind of functional group if that's a hydrocarbon on this side and another hydrocarbon on the other side? So we call that functional group an ester. What do we call that functional group where oxygen just has hydrocarbon on both sides? An ether functional group. Why wasn't there a chapter in our organic chemistry section about ethers as a functional group? Why didn't we learn about all the chemical reactions that ethers can do? Because they don't really do anything. They're very, very stable. We can hydrolyze an ether with really, really strongly acidic conditions. But ethers are much, much harder to hydrolyze than esters are. So, whereas phospholipids in humans have ester linkages that would hydrolyze at 70 degrees Celsius, archaea have adapted so that their phospholipids don't have that problem anymore. They use ether linkages instead, which are very, very stable at elevated temperatures. I think that's really cool, right? A fundamental difference in the phospholipid structure allows archaea to make phospholipid bilayers at temperatures where other human and other creature phospholipids would just hydrolyze. Don't you guys think that's a neat adaptation too? I think that's really awesome. Anyway, that's not in the book, but I think it's cool enough, especially given what we know about organic chemistry, that it's worth us taking a minute to marvel at a little. If you guys don't have any more questions from the lipids chapter, we will have tomorrow as a review day where any of the biochemistry materials that you want to talk more about, we can do that. All right. Make sure you bring your study guide for exam 3, and we can talk more about any of those things if you're wondering about like how much I expect you to know of those.
Can you explain again what you were saying about fluid mosaic model?
So is the fluid mosaic model in there?
That's the last question.
The fluid mosaic model means that the phospholipid bilayer is not stationary. The individual phospholipids are moving around all the time. It's essentially sort of like a lake of phospholipid bilayer. All right. Yeah, the individual membrane components are moving around. That's the
And
important.
that's for
thing.
any phospholipid.
For any cell membrane, we think about those components as a fluid mosaic.
Okay.
Does the Zoom contingent have any more questions for us?
I'm good.
I'm good too.
Okay. In that case, tomorrow is our review day and I'll see you guys then.
And then the test will be on Tuesday?
Test will be
Then?
on Tuesday.
Okay.
It's the plan right now.
Okay.
Have a good day, McKenna.